Now that we've kind of covered passive and active diffusion, and you can expect a 10-question exam on it, right? Because we've been promising that for a while now.
On diffusion?
On diffusion and osmosis and all the stuff that we've talked about thus far, that you've done a lab on, that we've gone through and answered questions. The next step, starting next week, we're going to have a lab where you're going to use the microscope to look at some things. Let's talk about the different kinds of cells, the stuff that we've talked about already. In essence, we have eukaryotic cells. Those are cells with nuclei. They have internal membranes. There are prokaryotic cells. They don't have any internal membranes. And then you have viruses. Viruses are the tiniest things. They're tiny. They don't have, most people, I should say most people, a lot of scientists don't even consider them alive. They don't have a lot of the characteristics of life. The first being, it's not a cell. A virus is not a cell. Although some do have membranes, it's not a true cell. Where prokaryotic cells are bacteria. So you see here, this is a eukaryotic cell here, a plant cell. This is a human white blood cell, the size of a human cell. And here's this tiny prokaryotic cell. This is a bacteria cell. Or this would be a virus. Tiny, tiny, small particles. And basically that's what they are. So when we're looking at nanometers, what does it mean to have nanometers? Everyone here is familiar with a meter, correct? A meter stick? All right, that's what 1 meter is. That's as long as a meter is. Take that meter and divide it 1,000 times into 1,000 little lines, and you end up with a millimeter. Obviously, a centimeter, a centimeter would be dividing that meter into 100 divisions, right? So when we write it in decimal, in an exponent fashion, 0.001 is 10 to the negative 3. Everybody have their calculator here? Their scientific calculator? Pull out your scientific calculator. If you don't have one, you should have it. It's part of your supply list. Open it up. The one, wow, okay. Go ahead and put. Let me see your calculator. Anyone, right here. There's different calculators at different ways. You're looking for 10 to the x, right? Not x squared. That would be squaring things. You have times 10 to the n. And this one says times 10 to the n. That's what yours says. Now, others might have different symbols, but it's times 10 to the n. So what we call it looks like this, times 10 to the n. Some might say times 10 to the x. So when you're talking about scientific notation, there's this, which is times 10 to the n. You'll see that on a bunch of different calculators, and some others you might have times 10 to the a or b or some symbol. The other thing that's actually very common is this symbol here, EE. On that red calculator you have there, that's what it says. It's in yellow. You see that it's in yellow? That means you have to push the yellow button before you push EE. It's like shift. The colors. May Lord Jesus Christ. So each color on your calculator requires you to push the color button before you push it. There's a main function and then there's a secondary function to each button. So if you see EE, that's the same as times 10 to the n. Is that clear to everyone? Yes or no?
Yes. So
if I have a number and I measure something as 243.6, something like this, and I want to turn this into scientific notation, what I do is I move the decimal to the ones place here. Now, what did I do to move the decimal from here to here? What did I have to do? I had to take 2.43 and multiply times 10 to the what? Second power, so that I could get 243.6. Now, why did I put 2.43 instead of 2.4360? Because I determined, and how we determine it it depends on a certain set of rules. I determined that there are three significant digits, three significant figures to this measurement, to this calculation. So 2.43, I only use the significant digits. I don't worry about the things that are not significant. Is that clear? So if I want to write this on my calculator the way you have there, I have to write, if I have the EE, I have to go 2.43 EE2. 2.43, that's what I'm pushing. Then I push the button EE, whatever it is I have to do to push that button. And then I would put in the numbers, 2. So 2.43 EE2. Do that now. 2.43 EE2. What'd you get? 243. If you do this by typing out this, then pushing that, and then pushing 2, you're going to get 243, because that's what this means. 2.43 times 2, 10 to the second power. Those of you that have 10 times 10 to the n, it's the same thing. All right, so why is that important? Because that's important because of what comes next here. Let's take a look at these numbers. These numbers this is 1 meter. Everybody here knows what a meter stick looks like? That's a meter. This is times 10 to the third power. So when we take that meter and we divide it 1,000 times, so 1 meter divided 1,000 little slashes, little marks, we call it a what? A meter stick divided 1,000 times. What is it what? Millimeter. There's 1,000 of these millimeters in 1 meter. 1,000 millimeters in 1 meter. So if you took a meter stick and counted each millimeter, there would be 1,000 of them in it. How many centimeters are in a meter?
In a meter?
100. Centi means 100. So centi. Milli, like a centipede. A millipede has how many legs? Pede means leg. A millipede is supposed to have how many legs? 1,000. It doesn't have 1,000, but they just call it a millipede because you have about. In any case, milli means 1,000. That's why 1,000th, right? A micrometer has how many? 10 to the negative 6. What is that number? 1, 2, 3, 4, 5, 6. 5 zeros before the 1. And you're right, it would be a millionth. A millionth, not 1,000th. Take that meter and divide it into 1,000,000 slashes. Now you have 1 micrometer. Now how much do you think of 10 to the negative 9 is? 2, 3, 4, 5, 6, 7, 8, 9. What is it? Which is it?
10,000.
100 million.
Somebody says 100 million.
A billion.
You think it's a billion? What if I took and I did put 1, 2, 3, 4, 5, 6, 7, 8, 9. That's there, there's there, and there's there. What is it?
That's a billion.
A billion, that's right.
Oh, somebody said.
Yeah, somebody did. But 100 million wasn't a bad guess. So it's good, but how do you figure out if you're right or not? This is science. We want our best guess, not just the
educated.
wanted educated is correct. Please be quiet, focus. So that's a billionth. So this is a billionth, this is a millionth, this is a thousandth, and this is one. This centi is 100 would be missing. So this is each one is 3 decimal places bigger than the previous one, right? So that's what we're talking about. 3 orders of magnitude bigger. These are scientific or international systems, SI units. You have an activity here you're going to need to use a ruler for. You're going to need to have a ruler. You're going to measure. Look at this big. Which one of these is biggest? Which is the biggest?
The Daphnia.
You think it's the Daphnia? Why do you think it's Daphnia? Look at them and tell me which is biggest.
Oh, wait.
Make a guess.
The Florida.
You think this one's the biggest? Not this one?
No.
Not this one?
Oh, that one whole thing. So it might be the infidel.
You have to count one. Don't count all of them. Just one. So
So this, but then what about the papillomavirus?
papillomavirus.
Yeah. Or it's just that small one right there.
This whole thing. All right. So what all of you are doing is you're not using the scales that are provided for you. You have to use the scale. So now let's talk about what that is. If I take and I draw a ruler out, I'm going to make a pretend ruler over here. And you have your divisions, you have your centimeters. Whatever. And your millimeters. What you're going to need to do is you're going to have to look at the scale. They give you something here. A distance from here to here is 1 millimeter. Millimeter. So I can take this and I put it here and I use this to measure it. And I said, don't change the size, obviously. You're going to use a ruler. So I'm going to say this much, whatever this distance is 1 millimeter. So how big is this Daphnia? 1 and a half. 1 and a half, maybe 1 2 3/4, whatever, right? Millimeters. Millimeters. Now I come over here and I line it up with this little thing here. And about half of this distance is a micrometer. So how long is this thing? Well, let's move it and find out. Let's see, there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 11 micrometers. Which is bigger, 11 micrometers or 1.5 millimeters? Right? And here's 100 micrometers. So if again, I'm changing the size of the ruler, which is bogus, but let's go ahead and say it's half a centimeter. Half a centimeter. So let's say 1, 2, 3 what? 300 micrometers. Well, obviously, if this was 10 micrometers, this is 300 micrometers. Obviously, this one's bigger than this one. But which one's bigger, this one or this one? Hard to tell because you have millimeters and this is micrometers. And then you have another one down here. These down here, this is micrometers, this is micrometers, this is micrometers. So again, but this is 0.1 micrometers for this whole distance. This is 50 micrometers for this whole distance, where this is 100 micrometers. So it's kind of hard to do. So this is what you can do. you can measure how many of these lengths you have in this picture. So you can pick the longest part and measure it on. How many of these lengths do you think are? And you could use a piece of paper, a cardboard, or a ruler, and say a number. Right? So like let's take a look at it and see. Let's compare it. How many of these lengths are going across? That's 1, 2, 3, 4. 3.5. You agree it's 3.5? So to be clear, when we're looking at this, and each one of these is a different size, the question they're asking you here is which one of these is biggest? And when you're looking at it, you just need something. You can use a card. You can use, look, I have a ruler here, but you don't really need a ruler. You can use anything. You can use an index card. Anything that has a straight line that has some kind of measurement. So if you use an index card, let's say, and you make a measurement from you take that index card and you move it, and you move this over to the scale. Do you see the scale? Do you see where it says 100 micrometers? That means everything that's this long is 100 micrometers. And so it doesn't matter what you use, but if you take it and you put it up to the line here, and then you mark it, you say, okay, from here to here is 100 micrometers. So you take it and you say, okay, well, that's 1, right? So that's 1 micrometer. And then you move it over again. Oh man, it wrote it on there. Okay. Why is it not erasing? I do not know. But okay, so that's 1, right? So again, from here to here is 1. So that's 1, then 2, then 3. Do you agree that's 3? So whatever, I think I said 3.5 a minute ago, but let's call it 3. How big is it from one end to the other? 3 what? Somebody said it. 300 what? Micrometers. That's this symbol means micro. So it's 300 micrometers. So this is 300. So that amoeba is 300. Ugh, I hate not being able to use my pencil, but let me zoom in here. 300 micrometers, right? So next one has a different length. So I'm going to use a different card. I'm going to take my card. I'm going to put it up on the length. You see it right there. I'm going to mark it. So from here to here is 1 micrometer. 1 micrometer. So that's 1. So I go to the end. I go 1. And I go ahead and follow it along. There's 2, and then there's 3, and there's 4, and there's 5, and there's 6, and there's 7, and there's 8, 9. It's not accurate, but let's say it is 9. So how big is that? 9 micrometers. So even though they look like they're the same size, one's 300 micrometers, the other one is 9 micrometers. That's this one, 9 micrometers. And you do that for all of them. So the problem is that there's some stuff in here that's not, they have this 10 to the negative 3. Do you see it? Do you all remember your scientific notations? In the previous part of this video, I mentioned scientific notations. You need to rewind it. I'll post it tonight. Rewind it and look at scientific notations and go over it. But What we know though is that a micrometer, if you look at it, 1 micrometer is equal to 10 to the negative 6 meters. So again, the question that they have for you to do, the thing they want you to do, is to take each of these measurements, these values that you calculated when you took a piece of paper, a card, and you lined it up, or even a ruler, and measured how many of these lines that you have here are found from one end of the creature to the other, one end of the cell to the other. And what you see is that you can, in the example we looked at with here at the amoeba, it was 300 micrometers. So now, how do we get to the millimeters? How do you convert these 300 micrometers to millimeters? We're going to use something called the factor label method. It is a standard method in physics and chemistry. This is necessary for you to learn this so that you're capable of converting any measurement into any other unit that you need to solve a problem. So when we have 300 micrometers, the way we set up the problem is we put what we have on the left side, so we have 300 micrometers, and on the right side of the equal sign, we put what we want, which is going to be millimeters. So next you're going to need a conversion factor, and to have a conversion factor, what you need is an equality, something equals something else. Well, we just got done saying that 1 micrometer equals 1 times 10 to the negative 6 meters. So 1 micrometer, sorry, I'm writing this in the car, so no, I'm not driving. 1 micrometer equals 1 times 10 to the negative 6 meters. Now, that still doesn't get us to millimeters, but we do know that 1 millimeter equals 1 times 10 to the negative 3 meters. So with this information, with these two equalities, these are equalities, with these two equalities we can make a conversion factor. And these conversion factors can help us get to our millimeter goal. So 300, as you know, every number is itself over 1. So we can go ahead and say 300 micrometers over 1. If we want to get rid of micrometers, we're going to multiply times something where micrometers is at the bottom. And if we're going to get rid of micrometers, I'm sorry, give me a second here. So 1 micrometer, as we said, is 1 times 10 to the negative 6. So if we place it so that micrometers is at the bottom, we can cancel micrometers and we're left with meters. But meters still doesn't get us where we want, because where do we want to get? We want to get to millimeters. That's the question. So how do we get there? We have to get rid of meters. Where are we going to put meters? Well, hopefully you sit at the bottom so that we can cancel again. If we multiply times meters, meters will cancel. So how do we get millimeters? Because that's what we want. We want on top. And get rid of meters. Well, we have an equality. 1 millimeter equals 1 times 10 to the negative 3 meters. So we're going to divide by 1 times 10 to the negative 3 meters. And that's going to give us how many millimeters. So you just use your calculator and multiply 300 times 1 times 10 to the negative 6. Divide that by 1 times 10 to the negative 3. And you get how many millimeters. Now the great thing about scientific notation is that, especially if you know your exponent rules, that you can do a lot of this calculation in your head. So if you are if you're multiplying exponents, you add them. If you're dividing exponents, you subtract them. So in this case, 1 times 10 to the negative 6 divided by 1 times 10 to the negative 3, you're going to add the exponents, subtract the exponents negative 6 and negative 3. So what is negative 6 minus, negative 3? Well, I hope you know that this means you're adding, right? Negative 3. So minus 6 plus 3 is minus 3. So what you have is 300 divided by 1, the micrometers have canceled already, times the meters have canceled, right? The only thing left is millimeters, is 1 times 10 to the negative 3 millimeters. Well, if you're 1 times 10 to the negative 3 means I'm going to move the decimal 3 times. In which direction? I'm going to move it to the left, because this is the same as dividing by 1,000. So I'm going to move the decimals 3 times to the left. So it's 0.300 millimeters. So it's 0.3 millimeters is 300 micrometers. So when you're looking at the calculation, this factor label method, and it's called factor label method because you're multiplying and you're labeling everything and you're canceling. And so it gives you a built-in checking mechanism so you can check your work as you go along. You knew you were looking for millimeters, you ended up with millimeters, and you see that everything's canceling correctly. So you're probably correct, assuming that you have pushed the right numbers in your calculator, and that solves a lot of your issues too. If you don't know can't remember the exponent rule or what to do with subtracting negative numbers, you can still do it pretty simply on a calculator. However, the great thing about scientific notation is that you don't even need a calculator, because you can just remember those 2 simple rules. If you're dividing by, when you divide, you subtract the exponents. When you add, and when you multiply, you add the exponents. And so your answers are self-checking. It's a great thing about the factor label method, and you'll be using that in chemistry and physics. So your answer for 300 micrometers is going to be 0.3 millimeters. 0.3 millimeters. 0.300, 0.30. Significant digits, when you're multiplying, dividing, there are rules. But let's just, for now, just say 0.3 millimeters. So you should be able to do the rest of these on your own and answer not just how long are these in micrometers, but then how many millimeters you have by doing the same calculation you have here. Now understand, you need to, in the very near future, you're going to need to be able to find these equalities in a paragraph or in a sentence or in an experiment, or you have to be given these inequalities somehow to find out what is equal to what. Then you're going to have to set up your factor label method, convert, and get what you need at the end. So you have to be able to do that very soon. So start to practice it. Use this as a practice on what you're going to need to be able to do in the near future. So list the 6 organisms in order by size. And as you saw, the problem with looking at this and looking at the size and saying, okay, this one's bigger than this one, obviously, is that they're all at different scales. So this line is smaller than this line, but yet they're equal value. They're both 100 micrometers. This one looks to be about the same as this one, but this is 1 micrometer and this is 100 micrometers. This one actually seems longer. It's certainly longer than this one. Seems longer than these two, and it's only 50 micrometers. So what you're talking about here is the zooming in, the magnification of the microscope is different. So the same length is going to is going to be equivalent to a smaller number at higher magnification and a larger number at a lower magnification. So you have to find a way to be able to convert these measurements, what you're seeing, into one standard, and the standard that's given here in this problem is millimeters. So before you can compare their sizes, you have to convert all these micrometer measurements that you're using this little line for, that you're saying, okay, well, this is 100 micrometers here, so how long is this? And this is 100 micrometers here, so how long is this? And this is 1 micrometer here, how long is this? As you saw with this example, this spirochete, you see that this we had 9 micrometers because it was 9 lengths from one end to the other. Now that was just a quick measurement. You could probably do a much better job at measuring that length, but in any case, 9 micrometers, converting it to millimeters, will allow you to compare whatever it is here to whatever it is here. Now, I did put 0.3 micrometers, but realistically I shouldn't have, right? I should have put, if I want to convert it to scientific notation, it would be 3.0 times 10 to, and it's hard to write, so let me zoom in. Again, I'm in a car doing this, so forgive the mess. It's 3.0 times 10, and it's 0.3, so it'd be to negative 1 millimeter, right? 3.0 times 10 to the negative 1 millimeter would be the correct way of writing it. Then you do that for each one of these, and as you do these, as you convert micrometers and meters, you'll be able to compare them, because not only do you have the same units, but you'll also be able to look at the negatives, and you don't have to worry about making a mistake on which decimal place you're at. So you can look at the coefficient, and then you can look at the exponent and make a decision on which one's bigger and which one's smaller. So list the 6 organisms in order by size from smallest to largest, and you can figure that out. Obviously, the bigger the negative, the more negative is, the smaller it is. And list those organisms. Study the scale of your ruler and state which of the 6 organisms you would be able to see with your unaided eye. So you guys can figure that out, and calculate the equivalent length in millimeters of the following measurements. Again, here's some measurements doing the same process. Please note, this is micrometer, so are all these, but this is not. This is in nanometers. So be very, 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 very careful when you're looking at these units, because it's easy to mix them up if you're not paying attention to detail. All right, you have a good one.